The Honorable Vice Dean for Academic, Research and Student Affairs, Faculty of Pharmacy, Universitas Indonesia, Dr. Apoteker Fadlina Chanisa Putri, Magister of Science. The Honorable Speaker for today's lecture, Professor Dr. Masafumi Muratani from Department of Genome Biology, Faculty of Medicine, University of Tsukuba Genome Biology Core, Transporter Medical Research Center. The Honorable Moderator, Apoteker Dona Mareta Aristanti, Magister of Science PhD from the Faculty of Pharmacy, Universitas Indonesia. The Honorable Professors, Lecturers, and all faculty members who attend today's lecture. And last but not least, I would also like to greet all audiences and participants. Thank you for joining us today. So the summer course is aimed for students and fresh graduates who are interested in improving their knowledge of pharmaceutical sciences with a broader perspective, because we invite professors and lecturers from Universitas Indonesia and of course well-known universities abroad. And today we will have the second lecture from Professor Dr. Masafumi Muratani. He will deliver a lecture about genomic analysis and methods used in COVID-19 research and space research. Before we start, I would like to explain the agenda schedule of today's course. First is the opening, second is lecture session, which followed by the discussion time. So there will be an hour lecture and an hour discussion. And third is certificate appreciation for both speaker and moderator, and then group photo session, and finally closing. Now we will move to our main agenda, the lecture number 2, which will be presented by Professor Dr. Masafumi Muratani and moderated by Apoteker Dona Mareta Aristanti, MSc PhD. Before we begin, I would like to read the CV of our moderator. Dona Mareta Aristanti is a lecturer in the Faculty of Pharmacy, Universitas Indonesia. Upon completing the Bachelor of Pharmacy and Apothecary Professional Program from Universitas Indonesia in 2008, she continued her graduate studies at the Tokyo Institute of Technology, Japan. She earned her master's degree in 2011 and doctoral degree in 2015. She was previously working as a postdoctoral researcher in the same university before coming home and joining the Faculty of Pharmacy, Universitas Indonesia, as one of the faculty members of the Laboratory of Phytochemistry and Pharmacognosy. Her interest areas are understanding the molecular pharmacology mechanism and biosynthetic of natural products. All right, without further ado, I invite our honorable moderator, Apoteker Dona Mareta Aristanti, MSc PhD. The time and screen are yours.
I'm very sorry for the technical problem. Thank you for the MC, Apoteker Risni, for the kind introductions. Good afternoon, everyone. I'm very happy to welcome you here today in this amazing event, the virtual lecture series from world-class professors about advances in pharmaceutical sciences. I am Dona Mareta Aristanti, a faculty member of Faculty of Pharmacy Universitas Indonesia. And today it is my great pleasure to be the moderator for today's topic, which is genomic analysis and methods used in COVID-19 research and space research, that will be given by Professor Masafumi Muratani from Department of Genome Biology, Faculty of Medicine, University of Tsukuba, Japan. And after presenting this topic, we will have the Q&A sessions. And then after that, we will have a 5 minutes break before Professor Muratani will have the second presentation about his current research. And also followed by another Q&A sessions. But before we begin the webinar, please allow me to read Professor Muratani's profile. So Professor Masafumi Muratani obtained his Bachelor of Science from University of Tsukuba in 2000. He then completed his PhD in Cold Spring Harbor University, Watson School of Biological Sciences, USA in 2005. He was previously working as research fellow at Cancer Research UK and also research associate at Genome Institute of Singapore. And his current research topic is technology development for genomics analysis of limited sample that we're going to hear more about it in his second presentation. Currently, he is working as a professor at the Faculty of Medicine, University of Tsukuba, Japan, and he also has non-academic professional experiences as a senior research fellow in Genome Science Research Center and also research advisor in Robot Biology Institute, Tokyo, Japan. He's also currently an active member of a Gene Lab Analysis Working Group, International Standards for Space-Omic Processing, and also Japan Society for Biological Sciences in Space. Such a remarkable profile, Professor. But before we move on to our main event, please let me to remind you that you are encouraged to ask questions throughout the webinar via the Zoom's chat box. So please type your questions at any time, and Professor Muratani will try to answer as many as possible during the Q&A sessions. All right, with no further ado, I'd like to invite our honorable speaker, Professor Masafumi Muratani, to give the lecture. Professor Muratani, the screen is yours.
Okay, hello. Oh, you can see my face. So let me share my screen first. Then, but before that, okay, maybe I share first. Okay. All right. So this is you can see my slide, first slide with pointer here. Okay, thank you very much for the kind introduction. And my name is Masafumi Muratani from University of Tsukuba. And I think this audience is, like a first time to introduce myself. So yes, as there was an introduction, I went to the US for PhD and then went to UK and Singapore, which is very close to Indonesia, and then came back to Japan in 2014. And since then, so when I was doing postdoc in Singapore, particularly, I switched my study to the genomics and then started to work on this genomics and epigenomics analysis of the limited samples. So limited means many things. Sometimes there is a low cell number, very limited amount of material, and limited sometimes like a precious. Like I work on a space research. The biological sample from space station is very precious, and sometimes the processing is very difficult. Limited choice of methods to use. And also, limited means deteriorated archive means some very old samples or some archive from the biobank and so on. So like many difficult samples, limited samples for the genomics analysis. And right now I'm working as a kind of rescue team. So if there is any difficult projects, we go there and they help to provide the analysis service or analysis and also data analysis for the project. And mainly these days I work on space research. And I run a genomics facility in which we process like a few thousand samples every year, including space, sometimes cancer research or different type of research projects. And also, of course, these days, coronavirus, the genome analysis of the corona is very important part. So using the same platform, genomics platform, we analyze the COVID research and also other clinical research samples. So at this point, I like to introduction. Maybe I can put some new slide here. So today I'm going to talk about the coronavirus COVID research, but is there any volunteer who can, say what is potential impact of the coronavirus in your, in your life, of course, but in your life science, biomedical research? Oh, I need to break this. There you go. Oh, this doesn't work very well. Okay, so I can see some. Oh, I can't see the participant. Okay, so maybe I can answer, respond to my own question, but when we experienced corona, genomics was one of the very important fields in, to work on the corona pandemic. And one of the topic was the genomics analysis of the coronavirus. And you probably experienced the PCR test these days, to test, and also as I mentioned, the coronavirus, the new strain of the virus, like a Delta or Omicron, these are all based on the genomics analysis of the coronavirus. And all these things are related to the genomics. And today I'm going to talk about the how this coronavirus research is related to the genomics using this paper as an example. I just put the link to the Google Drive, including this paper and today's presentation slide. And this is okay. So if you have any questions, please let me know. So in this paper, the pneumonia outbreak associated with new coronavirus probable but origin use this. Really is the first report of the coronavirus genome structure and sequence, and published in 2020, but it came out online in February or something. And then did multiple analysis of the coronavirus. First, they used the clinical samples from the potentially infected people, and then identified this virus is something similar to the previous SARS virus. And then did identify the genome structure and compare the genomic sequence, the similarity to the previous coronavirus, and also the bat SARS virus. And then did a phylogenetic tree to identify which virus is similar to the current pandemic causing virus, and detection of the virus by PCR. And then this is the antibody test. And also they tested infected to the cell line model to identify potential receptor. So this paper includes lots of genomics analysis. So I am going to use this example to explain how coronavirus pandemic was first analyzed using genomics technologies. Okay. All right, here I go. So the bioinformatics in genomics, so data analysis in high throughput sequencing, and main topic is this coronavirus study. But before starting to do that, I will give a short introduction about the genomics. And then this first part of the lecture, I will explain some of the technologies used in these publications, which is relevant to the coronavirus. And after this part, we will come back to Q&A, and then if we have enough time, we will go through this some research examples from the Tsukuba Genomics platform. Okay, so firstly, I like to start from this slide. Okay, so the collection and analysis and visualization of the data is very important in this modern society. So this is an example of the GPS, the global positioning system. In using this system, you can identify where you are and what you are doing right now. And then in a more research-oriented examples, imaging is very important these days. So this is a part of the Japan and representing the temperature in the sea water. And without going to this, the around the area, by satellite imaging, you can detect the water temperature and potentially visualize the current and so on. And in this example, so the satellite image is just a scanning of the maybe infrared light, but using this visualization of the data, you can see the like a location and the distribution of the temperature of the water. This is another example of the Pacific, across the Pacific Ocean, and also during the different time of the year, the water temperature change is visualized. This is another example of the rain cloud. Without going to this location, you can measure and detect what's going on in the rain cloud. So the same thing is true for genomics too. In genomics, the data collection is a genome-wide. In human genome, there is a 3 billion base compared to this Earth, size of the Earth, which is 5 million square kilometers. So if you think about the scale of this 3 billion to 5 million, you can see how big area you can potentially explore using the genomics technology in a genomics research. And also for the data analysis, we are eventually trying to do system level analysis. This example is one of the previous generation of the fastest supercomputer in Japan, the K, which used 80,000 CPUs. But if you think about the human body, there are 37 trillion estimated cells, and each of the cell has more than 10,000 transcript variant for each genes from the entire genome. So the complexity of this, so many cells, so many genes are controlled under in response to the environmental stimuli or developmental stage, then that creates the complex human body behavior. So eventually, this system level understanding of this very complex creature is our goal. So for this data collection, how to collect the data is this ultra high throughput sequencing, which came up around year 2008, 2007, which is called next generation sequencing at that time. So the important feature of this high throughput sequencing is there are many, we can read many large number of the DNA molecule at one time. So this is image, one of the image of the flow cell. In this clear area, there is a glass plate with the liquid, like a channel for liquid to flow. And by applying the DNA liquid here, each individual DNA stick to the glass surface treated with this probe area. And then essentially each individual DNA molecule is amplified by solid phase PCR reaction. So amplifying single molecule into the large number of the clones, which makes the cluster. And then by incorporating the different fluorescent nucleotide, we can visualize the DNA sequence by the imaging. So incorporation of the ATGCs, the different color of the nucleotide can be detected by imaging. And nice thing about the genomics is you can read DNA and RNA from any species. As far as we know, essentially all the creatures, the organisms on the earth share the common DNA and RNA-based genome structure, which creates the proteins and do all the activity, life activity. And wherever the sample comes from under the ice or sea or human or some plants, these can be done, analyzed using the same platform of the DNA sequencing. And then once you know how to prepare the sample, read the sequence, and then do the bioinformatics analysis, you can actually apply that skill to analyze any sort of domain on life. And then the next question is how to analyze the data. As I mentioned, the point of the data analysis, so original data is lots of ATGCs, but to process the data, the final goal is how to visualize the data so that people can understand visually. This is one example of the Sarcos plot from MCF's breast cancer cell line, showing a lot of the DNA, the chromosomal amplification and deletions, and also this kind of line, the structural change of the genome, the fusion and translocations. And this example is biological pathways affected by the different the differentially regulated genes and how these genes are functionally connected. And this is the example of the visualization across the gene location and then many events, for example, like epigenetic mark of histone modifications. This is DNA methylation. And there are some feature of the DNA binding protein locations. And here is a motif analysis. And of course, you probably see a lot of the heat map in the gene expression analysis in the publications. So these are kind of like a genomic analysis overview. And then I like to explain how this genomics technology is actually used in this recent corona pandemic in a COVID to respond to COVID pandemic. Yes, anybody say anything? If you have any questions so far, I'm going to talk about the how the genomics is working very important, playing important role in coronavirus. But what kind of field are you working? Like you are like a department of pharmacology, right? Any like maybe one or two volunteers to talk about explain like how the COVID affects your research topic or interests or anything? Maybe I can see. Any volunteers? How many people had corona antibody tests so far? You know, immunology. Like, can you raise hand?
So the participant is pretty shy, I guess.
Okay.
I think most of us in Indonesia are, at least once. Oh, there's this one participant response in the chat box.
Oh,
Professor.
Chat. Ah. Okay, this pandemic had pushed the implementation of NGH machine in my workplace. Yes, that's great answer. Okay. Yeah. Okay. Vicken-san, can you speak up? What
Yes.
kind of workplace
Hello?
are you in?
Can you hear me?
Yes.
Currently, I'm working at a biomolecular laboratory.
I see. What kind of are you working on any genomics analysis or like a PCR test or anything?
Mostly we use PCR, but currently we're preparing for tuberculosis research.
Oh, I see. That's very good answer actually. So, the corona just suddenly came out, but actually many genomic analysis platform was already existing. So, since this, beginning of the pandemic, many activity in the genomics facility shifted to the COVID. So, I think you mentioned about this genomics analysis platform and then did a PCR, but maybe previously there are many type of the assays, but right now the current priority is the corona, and also particular the strain specific PCR analysis might be important. So okay, thank you very much. That's very good answer. All right, so I come back to my slide. Okay, so I will go through some of the techniques used in this COVID paper. If you haven't looked at the paper, please, I would recommend to read this Corona paper, because this is, it's probably in a once in a lifetime situation and impacted to the many people's life. And if we are working on a biomedical science, maybe it's worth, knowing maybe towards the future, it's going to be a history of how this biomedical science coped or responded to this very important topic. Okay, so the first page of the paper is introducing this, the outline of the paper, summary page, but one of the things they mentioned was this full-length genome sequence was reported in this paper. And I think, so that's full-length genome sequence. And also, if you go to the method section, there is a metagenomics analysis using next generation sequencing, blah, 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 and by de novo assembly and targeted PCR, and then remapping of the total reads to this genome, blah, blah, blah. So there are many words using genomics technologies. So I like to go through these kind of keywords and then explain what these assays means. Okay. So the first topic is next generation sequencing. I just explained about one example, but again, this slide, and I like to explain how this typical high throughput sequencing analysis will go. And so as a kind of brief overview or principle of the genomics next gen high throughput sequencing is this making cluster on a solid phase and then doing the sequencing by imaging. But if I explain a bit more detail, this is the first step of the sample preparation. So this slide explains from sample to data overview. If you haven't used or experienced the high throughput sequencing yet, but you probably are familiar about this extracting DNA and RNA from the maybe tissue specimen, or in the corona case, it's kind of like a nasal swab or saliva samples. And the first section is extracting RNA and DNA, DNA isolation. So this example shows kind of a cancer genome analysis from the FFP tissue block. There is a slice, and then FFP slide, and then scraping this slide of the tissue, you extract the DNA, and sometimes as a control, the whole blood DNA is extracted, and then here the genomic DNA, and then this DNA or RNA is fragmented into the small pieces, and then put the adapters and so on and amplified. And this process is generally called library prep and sometimes capture or enrichment, but library preparation. And then this library is put into the sequencing machine to obtain the data, and data will be eventually analyzed by computer. And next slide will break down this 3 processes into the further. And the important thing about the genomics these days is the name called library. So if you think about the library, it's kind of like, the lots of books in building, you read many books in the library, but when you talk about the library in genomics, this indicates the collection of lots of DNA and RNA fragments. So this is an example of the RNA sequencing. Coronavirus is also RNA virus. So the initially isolated RNA is usually randomly primed to convert this RNA into the complementary DNA. This is also same as the coronavirus. And then putting the adapter on both sides of the this original sample sequence. And then eventually this adapter ligated DNA fragments or cDNA fragments are amplified and then put into the sequencer. So at this point, the library is very important word. So this is material converted the original nucleic acid, DNA and RNA into the DNA fragments that can be read by the sequencer. And then once the library is prepared, this library is usually this sequencing reagent come into the cartridge. We put this sample into one of the holes and then put this machine cartridge into the machine to start the sequence. And inside of the sequencing machine, there is a kind of like a treatment to stick to this adapter sequence. And then using this same adapter sequence, use this as a primer to incorporate the nucleotide one by one so that you can sequence by imaging. And then by looking at the image here, the blue nucleotide is incorporated, and then we can call base as a T, and then the green, the G, the red, the C, and then again the blue T. So using this synthesis cycle in the machine, you can read the sequence one by one. And then there are many different platforms. And the first, like raw data from sequencing machine is always called FastQ5. And the FastQ format consists of this 4 lines in one read. So the first line is the name of the read. And then second line is ATGCs, the sequence. And then third line is currently the placeholder. We put plus. And then fourth one is a quality score. So this like lots of text characters. This indicates 40 grades of the how confident these base calls are. So, and then this is one set of the sequence, and the next one. So this lots of sequence reads are stuck into the text file. That's the FastQ file. And this FastQ file, I just mentioned about the quality score, and the quality score represents this 40 digits of the how confident these base scores are. And sometimes this base score is quality score is important because towards the end of the cycle. So in these examples, from first cycle to 75 cycle, so reading the 75 nucleotide in the machine, the quality score comes down, which means from the beginning towards the end, the sequencing reaction become less efficient towards the end of the read. So sometimes we cut the sequence raw data based on the quality score. So that is done by this trimming of the sequencing read. So in this Corona paper, this paper used a tool called Cut Adapt. This is a published tool available in, you can download the sequence, the program, but also it's available in Galaxy platform, the cloud computing platform. And there is this is just an example of how usually these published tools are used. If you go to the website of the Cut Adapt, there is a usage and how to install, installation and user guide. And one by one, bioinformatics is very helpful because everything is described in this kind of like a user guide page of the tool. Okay. All right. So then, based on this explanation, you can set the parameters how you want to cut the adapter based on the score, quality score, or based on the length. And this cut adapter adapt tool is also in the cloud computing platform called Galaxy. If you go to Google search Galaxy and Genome, there is a Galaxy website which has many genomics analysis algorithm or tools, analysis tools in a cloud format. So when we do the bioinformatics course in our university or in other universities, we typically use this Galaxy platform. So this is one thing like you probably want to do the internet search and see how it works. And if you are interested in, you can join to any open course or Tsukuba course potentially, so that you can experience how to use it. And the point is that you can start using many informatics tools without typing the commands. So maybe most of you are working on the wet lab or may not have the previous bioinformatics analysis experience, but it's actually very easy to start. And the data is usually freely available for many practice use and also research use. And here is the original article of the analysis tools, like your COVID research paper, and then doing internet search of the tools, or you can go to Galaxy for many of the major tools, and then start using, try to how to use the experience, how to use these tools. So this slide is uploaded to the Google Drive, so please have a look later. And then how to sequence genome. So in this paper, they used both mapping method and de novo assembly. And I like to highlight these two different strategies of how to determine the sequence. So firstly, the mapping is if you know the difference sequence, which is when you work on the human, mouse, very like well-established organisms, there is already a standard genome sequence, which is a reference sequence available. And by mapping your sequence, you can detect the difference between your samples to the reference sample sequence. So typical application is the mutation detection. In coronavirus, there are many substrains, Omicron, Delta, or Alpha, Beta strains. These are all depending on the how it's these substrains acquired mutation compared to this first coronavirus reference sequence, which is actually determined in this publication. And then another thing is when you don't know the reference, this paper first isolated the coronavirus, new coronavirus in this COVID-19 pandemic. And, if initially people don't know the sequence, but they successfully assembled the sequence, genome sequence, which is a de novo assembly. So typical application is determining the new genome. So I like to explain these two different types of genome sequencing technologies. So the first thing is a mapping of the genome sequence leads to the reference genome. So if you have a kind of like a standard reference genome sequence, like a human genome, and then you have a sequence lead, which consists of the kind of like opposing direction of the short lead on this side and on this side. And usually the high throughput sequencing lead is relatively short, like a 35 base pair or 36 base from both sides. And interestingly, even for human genome, this is 3 billion base pairs. If you know the 35 base and the two pair, like one pair of the 35 base, and you know about the distance, and then orientation, looking at the inside to sequence, then you can essentially determine the unique position in the genome, except for like a telomere or repetitive sequence regions. But most of the genome, you can identify the single location, which you can map, identify where this DNA fragment came from. And then again, this mapping tool is available in the Galaxy interface. This is the cloud computing platform. And the next one is a de novo assembly. How to identify the corona genome without knowing the sequence. So this does not use a reference, and how to do this. So let's say here is the original genomic DNA, or in corona case, it's RNA virus, and then randomly fragmented into the DNA or complementary DNA of the coronavirus genome, and then do the lot of sequencing, and then assemble the sequence. So how to assemble the sequence? So this is a kind of basic principle of how this computation will go. So this, let's say this is the original genome sequence. And then if you sequence this randomly chopped fragment, you probably get lots of fragments which represent part of this entire genome. And then computationally, you will search for the similar end, and then which is assembly. And then stacking up the similar DNA sequence leads, like this cartoon, like, oh, this GGGG is similar to these guys. And then this fragment is covering a bit more upstream, but this next fragment that has a similarity to this fragment is extending more towards 3' and so on. Then by combining lots of like similar ends, people can eventually identify the entire genome sequence. So which is very, looks like very complex, but again, this de novo assembly tool is available in as a research tool already. And then this slide is a bit complex about this, how evaluating the quality of the assembly, but let me skip. And then shotgun metagenome sequencing is here. So let's assume there is a mixture of the genomic sequence. In this case, in the corona case, there are a large amount of human DNA and RNA, because it's kind of like coming from the lung washing liquid. Then there are some different type of the microorganisms, and also lots of coronavirus genome. And then in reality, this many genome cDNA sequence derived from the many human and pathogen genome is mixed here. And then people do the shotgun sequencing. And then here is assembly and alignment with genomes reference in database. So particularly in corona case, there are many human RNA sequence here. So first you map into the human genome and then identify non-human sequencing lead. And then using this as a de novo assembly for the de novo assembly. So in this, in such a way, coronavirus sequence was determined. Maybe I can go back to the, okay. So let me go through. And then this MegaHit is one of the metagenomic assembly tools, also available in this Galaxy interface. And then this is just summary of the genome sequencing by short-read sequencers. There is a classification, the whole genome shotgun selection method. So this is sometimes the whole genome is not very kind of like a cost a lot of sequencing read. Then people use capture and amplicon to enrich the target region to read. So one example is capture is exome sequencing, only captures the gene protein coding region of the genome. In this way, people can identify the cancer-associated mutation very efficiently. And amplicon sequencing is another way to enrich the target of the sequence by performing the PCR amplification of the important place and then do the sequencing. This is exome. This is amplicon sequencing. So in coronavirus, the corona genome analysis, this amplicon sequencing is used quite often. So this is an example of the, yes, this is a 29 kilobase. So this is actually coronavirus genome. This coronavirus genome is covered by about 100 amplicons. So if you do the close-up view, this primer set, primer pool 1, amplify this using this and this. And then this also contains non-overlapping primer sets. And then primer set 2 contains slightly overlapping primer set, and primer sets pool 3 amplify slightly again like a different region. And in coronavirus genome analysis, we typically use the 2 pools, pool 1 and pool 2, to amplify all the regions of the coronavirus. And then through putting this PCR product into the sequencer, and then do the corona genome mutation analysis. And this is this is coming from the exome sequencing, but this is the typical appearance of the sequencing mapping. So in this case, this probe capture region is trying to read this exon regions. And as you can see, towards this targeting this genomic area, there are many sequencing reads. And sometimes this vertical bar appearing, this is variant code, which is different from the reference sequence. So by doing so, we can identify potential mutation, which is difference compared to the reference sequence. And in the same way, we can analyze the coronavirus genome to see which mutation is included in your sample. And by the close-up of this mapping, here is how this base code look like. So there is a TTCGAG, that's a reference sequence, but sometimes here is A, like TTCA, AGAA. So this variant difference from the reference sequence is highlighted in the red color. And this T might be just a sequencing error because this particular lead has lots of difference compared to the original genome sequence difference. So could be a wrong mapping, or this fragment is coming from very highly mutated cells potentially. Another advantage of the high throughput sequencing is to Sanger sequence is this base core is very quantitative. So if you do the Sanger sequencing for the mutation analysis, particularly for somatic mutation, you see this kind of like a wave of this CACCTG and so on. But maybe like sometimes you see this minor curve. Maybe this is a mixture of G and T. Actually, C and T are here, and this is the T and C double peak, which you can identify as a SNP or somatic mutation. But this kind of like a small peak is very difficult to call the base call. This can be just a sequencing error, or this can be a real mixture of the minor allele. So in such case, the high throughput sequencing is very provides a clear-cut result. How many A's and how many C's containing leads are there, and also when there is insertion and deletion, which destroys the phase information of the Sanger sequencing, the high throughput sequencing is very powerful to cause the mutations. Okay, so this is some discussion about the sequencing coverage, about to evaluate the data quality, but let me skip this slide. And then this is kind of like a brief introduction about shotgun sequencing and targeting sequencing and mapping and de novo assembly, and also base coverage and physical coverage, the quality of the data, sequencing data, and metagenomic analysis. And today we only use a short read for coronavirus, but there is a many advantage about long read sequencing too. So at this point, I like to go through this paper just to kind of like remind actual example of how these technologies are used. Firstly, this full-length genome sequence was obtained from 5 patients. So in abstracts, they mentioned about obtaining the full-length genome sequence, but in the method section, they explained a bit more detail, combining different type of sequencing. And then this figure 1 shows metagenome analysis of the lung washed liquid, BALF. So first, they isolated the RNA from the clinical sample, and then removed the human sequence first by mapping. By mapping to the genome, there is a non-human sequence which left as an unmapped sequence. So then they draw the pie chart of this composition of the unmapped sequence, and then they compare the similarity to the known genomic sequence of non-human pathogen maybe, and then identifies many sequences were similar to the SARS virus, which came out. It's already 20 years ago, nearly 20 years ago. I think around year 2000 or something, there was a SARS virus. It's described in a paper, sorry. And then because of the similarity, they suspected this new pandemic is caused by something similar to the SARS virus. Then they performed the de novo assembly of this using this lead, and then identified this genome structure. And then next, they compared this new coronavirus genome against various types of the coronavirus, including SARS virus. So the SARS virus similarity is about maybe around 50% to 80%, depending on the region. And then they identified even closer virus genome, which is isolated from bat previously. So that's a major supporting the major conclusion that this pneumonia pandemic is caused by the virus that is similar to the SARS virus, and then even closer to the other isolated virus isolated from bat, supported by the genomic, the phylogenetic trees. And then eventually, after this publication of the paper, which identified the bat similar virus, more and more analysis came out like this, more similarity or detailed comparison with different virus, and then more lines. So this complex version of this figure is published by many other papers comparing many virus. Then different origin of the virus might be proposed and so on. So that's how this coronavirus, the origin, hunting for origin was still, I think, ongoing. And then another thing about this genomic analysis is identification of the substrains. So these days, the Omicron strain is widespread, but before that, there was a Delta, Beta, and Alpha strains. These substrains are all tracked based on the mutation in coronavirus genome. And if you go to the website of the next strain, this is again originally set up as a framework to monitor the flu virus, but because of this corona pandemic, it suddenly redeveloped or reformatted towards the coronavirus strains in the worldwide. This provided the early alert about what kind of substrains are coming up, isolated, and then indicating potential geographic location of the spread of this virus, helping the decision by the government and health authority to how to predict the spread of the new strains, and also the distribution, allocation of the medical resource to cope with this pandemic. And this is these days more and more strains coming up, but in about 2 years ago, the situation was relatively simple. People called the substrain based on the geographic location, but these days it's more like a unified idea about this the classification of the substrains based on the number and also some Greek alphabet. And again, just to emphasize, this analysis used in this paper are actually all available in this cloud computing platform, which anybody can register and use. And original data from this coronavirus might be some restriction only for academic use, but many published papers, even for the corona substrains, are available in a public database. So if you are interested in, please try to open this link and then explore how we could potentially use or try informatics analysis. Okay, so final slide of this part, the first part of the lecture is just advertisement in University of Tsukuba. We provide this bioinformatics course for the biomedical graduate students in which we experience for the first time users of the bioinformatics, this genome browsers, web tools, Galaxy, and how the Linux, some command line, and how to download the public data are available in NCBI or EBI or DDBJ. So these analysis will be broadly used in thesis research in a graduate program as research study in Tsukuba. And also it's probably similar situation for any part of the world. So if you are interested in, please try. There is some textbook available too. And this is just an example of the before the pandemic, just before the spread of the coronavirus, I actually visited Jakarta for 2020 January course. So similar course can be done. The computational resource should be there. So it's encouraged to try. Okay, so that's first part of the lecture. So any questions, comments? Maybe I cannot see the face or screen, so let me break the, or maybe I can go back to.
Okay, thank you, Professor Muratani, for the very intriguing and insightful presentation. So, students, ladies and gentlemen, let's start our discussion sessions for the first talk from Professor Muratani. Everyone is allowed to ask questions, and for the Zoom participants, please just use the raise hand feature or just type your questions in the Zoom chat box. Okay, let me check whether there's questions from the participants. Okay, there's no questions yet. So waiting for the questions to come in. I have actually quite many questions for you,
Yes, please.
Professor. Well, it's such a long process, and it's a little bit intimidating, to be honest, because I'm not in the field of genomic, and it's do you do this on your own? I mean, like in your lab, or you collaborate? And do you have any tips or like recommendations, or what should we do? What should we think? What's the first step to do if we want to try doing this kind of research?
So I think there are two parts, like wet lab and also dry lab.
It will be in that one.
And then for maybe you are like a university, like a Tsukuba too, like to implement the wet lab like analysis platform. Since somebody mentioned about the introducing sequencing machines, so that's actually enough set up. So usually for research purposes, we continue to run the genomics facility, and then sometimes there is like an occasion to use this genomic analysis for the like a corona analysis or cancer studies. And as I mentioned, the genomics is quite common from the different creatures. So all use the DNA and RNA. So your school is a school of pharmacology, so mostly like a medical school, but that resource is also useful for the other domain of the biological science. So to increase the usage of the machines, if we analyze more samples, we can buy more reagents at lower cost. And also we can do more practice using the different samples. So keep the facility running is very important for the wet lab. So gathering more interest for any sort of domain of the research is good strategy to do the analysis. And once somebody knows how to do the analysis of RNA and DNA, many things are common for the wet lab process. And then next thing is dry informatics, but I think that bioinformatics is more easy to set up, and for the institutional level and also individual students and researchers, because data is most of the data is available in the public domain. And also it doesn't cost, like reagents and many experiment is very expensive to perform, but data is, whatever you do the analysis, original data is in a database and you never destroy it. And also it's free, you need to pay the electricity and your maybe laptop computer, but using this Galaxy platform, cloud computing platform is free. And you can upload many data and then store actually a lot of allocation for each individual user. So if you are interested in, please, like you can start anytime. And if you have any questions, usually these bioinformatics tools, difficult part is everybody is same. So somebody should have the same question previously. And then if you go to Google search, the error message you see on your display or some sort of like, the keywords, there is always some answer. There are many people wondering about same question. So you actually don't need any teachers. If you have internet connection and relatively small laptop computer, you can start. So it's up to like how much effort you want to spend. And then for my case, I started from wet lab, like a pipetting. And when I moved to Singapore, I started to join to the Genomic Institute. So that's an opportunity. But at that point, the high throughput sequencing just came out. So I started to learn many, several, like attended some course or the typical bioinformatics course for the wet lab people. And for the first several times I failed, I never get used to this types of command line. And I also didn't understand the mathematics behind this software. But when Galaxy came out, it was very easy for me to start. So that's the first time I practically started to understand and also started to do the analysis I wanted to do. So again, like Galaxy, I would recommend. Now it's a lot of resource there, and many, even for this Corona pandemic research, all the resource tools used in that paper was actually available as a pre-installed software in the Galaxy platform. So I would recommend to start from that point, I think.
Yes, very interested. So this Galaxy tools, is it free? I mean,
Yes.
like for it can be accessed by everyone.
Yes.
Like
If
all
you-
free. You
Yes.
For example, if you want to analyze a more detailed or a more advanced analysis, you have to like pay, no?
No.
Okay.
Actually, this is supported by, I think, U.S. government.
Oh,
And also,
okay.
there are
Very
many
interesting.
mirror sites. Also, some of the Japanese institutions support the Galaxy on their own site, but it's all free. And data is also, unless you handle this, the human genome data, which is should be protected, but your account data is your own. So even for me, I often use for quick analysis of the practical a daily research activity, I use Galaxy platform, and also to send the file to my collaborators. It's very convenient platform to share the data. So I upload the data and then pass that data to our collaborators.
Okay. Very, very interesting. So is there any questions from the participant? This is very interesting for me, but I have to give the chance for the participants to ask questions. No questions? Oh, okay. For Delin, Novifa?
Itu sama yang hasilnya nano itu, diameter. Enggak.
Yes, you can ask.
Yes.
Ya, memang alatnya satu itu deh. Cuman kayak
Preparasi.
Thank you for the opportunity. As we know that COVID-19 is RNA virus, right? So it's easy to mutation. Based on your experience, is there any mutation of SARS-CoV-2 that can benefit for people, or maybe the other way around?
Oh, the mutation in what?
In
I
SARS-CoV-2.
Oh, you mean SARS virus?
Yes, yes.
Maybe I don't have enough knowledge about this previous SARS virus. So yeah, I'm not really like a virology person. So like each individual mutation impact is I'm not very like I'm not understanding very well. But we do provide corona genome sequencing analysis to provide the analysis result to the more like the National Institute controlling the infections. Maybe. Yes. Maybe next question.
Okay, next questions for Lee. How do you pronounce it? Lee Nguyen Long.
Hello, can you guys listen to me? Hello?
Yes, yes, loud and clear.
So I wanted to ask that if there is a sample of DNA or RNA that's beyond recognition due to deterioration by many factors, is there a way that is cost-effective to resequence it in smaller labs and even field labs?
Okay. I think one way to analyze like at low cost is these days the sequencing capacity itself is so one solution is to send the sample to the analysis lab, which is usually faster, and also for us maintaining like a purchasing machine and maintaining the facility is quite expensive. And also, as you may know, the sequencing reaction or like a sequencing run, you can mix like many samples by putting the barcode. So for our facility, like increasing like a few more sample is not particularly like damaging. And also for the workflow wise, like a 50 sample and a 60 sample is not much different. So one thing you can consider is to just ask nearby collaborators or friends to read the sequence for you. In small lab, yeah. And also there are some Illumina or other companies sells a smaller output sequencer, like a minisig or that sort of like a smaller sequencer. But again, like the library preparation, as I mentioned, is one more step you need to consider, and then to set up the routine work for library preparation and quality control, like checking if your library is good or not, is also you need an extra layer of the equipment, and also if you are not doing routinely, you need to do the practice run, or you might fail. Then that's a lot of headaches. So it's probably like nice to work on a data analysis side, at least by yourself. Then the wet lab side, you can find like a nice genomics facility as a service, I think.
Okay. There's one other questions in the chat box. So I think she asked how to differ the virus when you read the sequencing, depending to where it came from, and how many samples that you have to represent a specific variant, and to decide which specific variant when there's an outbreak?
That's a very good question. I think in reality, there are so many variants, and then Delta Omicron is really is a major population of the distinct virus population. And when we when really first several, maybe 10, 20 cases came into Japan, and then identified around a country, and then that data actually had a very distinct one or two mutations that we can distinguish the isolate from Tokyo and isolate from Kyoto area. So that resolution, I think the coronavirus is mutated everywhere every day. So yeah, practically I think every infection case could be different, but the major strain is isolate is impactful mutation will be expanded very fast. So that is called as a major strain in a news report and everywhere. So if you are actually working on a genomics facility, there are so many scattered spontaneous mutations we see. That's diverse. And also that's the case for the cancer genome too. So if you analyze the cancer genome mutation, the EGFR or KRAS, there is a distinct hotspot, but that means throughout the life of the cancer development, every single nucleotide is tested to mutate. So that's why we isolated really the same nucleotide, same convert, change of the base substitution we see. So it's very established mutations are highly selective, and I believe there are some advantage about how the spread of the corona.
Okay.
I hope that answers. Yeah.
Okay. Is there any questions? Other questions? One more questions before we have a break, and then Professor Muratani will have another talk.
Yeah, another part is a bit shorter, so
okay.
Don't worry.
Okay.
But if you have any questions, we can also come back to this first part.
Okay. Okay, I have a questions actually,
Yes,
Professor, for the metagenomic analysis, because in metagenomic there are a variety of microorganisms that can be found, right? So how do you do the grouping? I mean, if you have the data, is there any special or recommended application, for example, bioinformatic application, how to do the grouping without, usually you don't do the grouping manually, right? Or you do that?
Now, so this like a tool, like a mega hit metagenomic tool, usually use the database of the known strains. So usually bioinformatics is because original data is lots of ATGCs, right? So unless it's highlighted by software or something, it's something like not visually recognizable. So we never know what we are missing actually. There are lots of leads that are not mapped, And then even we do the de novo assembly, most of them cannot be assembled nicely. So that's something like without somebody isolate and put in the database, usually it's very difficult to recognize what it means.
Yes.
So even human genome, there are many unmapped leads, and it can be some hidden pathogens infecting to human. We don't know. There could be some sort of like a very strange region of the genome which exists in the Japanese population but not in the reference. It's potentially possible. So the one thing we should remember is because this informatics tool is original data is not recognizable for humans. We never know what we might be missing in the leftover data.
Okay. Okay, thank you for the explanation. Is there any questions before we take a break? Okay, I think there's no more questions for now, but we will have a second Q&A after your talk. So the participants can still type your questions, and then Professor Muratani will try to answer as many as possible. Okay, so we're going to have 5-minute breaks before we're going to hear Professor Muratani's talk, the second talk from Professor Muratani. Okay.
Yeah, thank you. Some break time.
Yes, Professor.
Becoming a pharmacist is my destiny. Being there for people is a must in this job. But it is not that simple. It all started from Faculty of Pharmacy, Universitas Indonesia, the place where I learned. Everything that I learned at the Faculty of Pharmacy, Universitas Indonesia, keep inspiring me to be forever dedicated.
Okay, welcome back, everyone. I hope you've refreshed and ready for another talk from Professor Muratani. So Professor Muratani will have the second talk. Without further ado, Professor, the screen is yours.
Okay, so let me share my screen again. So as I mentioned, so we are working on the a limited sample analysis, and this technology is very useful for precious sample too. So that's how we came to the kind of Japanese Space Agency. In the town, we have actually two very prominent rockets, one standing up and one lying down. And there is the satellite, a testing facility, and also astronaut training facilities. And as you may know, so SpaceX, like a Blue Origin, or there are many companies, private companies these days are developing the spaceship to send the people to the low Earth orbit or moon, and even trying to put the people into the Mars. But the hardware development is quite interesting, making rockets and spaceship. But there is also a big problem about the life science in space. So the rocket can travel to Mars, the round trip 2 to 3 years missions. But currently, it's a big problem about how to let the people survive during this long duration trip. So there are space radiation and microgravity impact. So even in a low Earth orbit, the astronauts experience many potential health problems. And currently, the longest stay in the space is about a bit more than 1 year, but towards Mars, 2 to 3 years missions minimum, how to survive is a big problem. So we are working on the mouse experiment to learn about the molecular mechanism in the space. And also I lead the liquid biopsy project from astronauts. So our lab is working on different projects. Some people working on the cancer, some people working on space, and so on. But majority of the activity these days is space research. So this is but if you go to the microgravity environment, we know that there is muscle and bone loss happening, and in the recent years, we successfully reproduced that, launching mouse and keeping mice in the space for about 1 month. We observed, as expected, the muscle atrophy and bone density loss, just like human. This is a mechanical unloading phenotype. But also, we see many different types of the reaction, which previously not expected, like just not muscle mass, but what muscle fiber type is different in space, like immune system, metabolic change, and oxidative stress is different. And also there are different neurological changes, including circadian rhythms. And also our recent research to space research showed that there is intergenerational effects of in mouse. Essentially, if you fry male mice, that sperm is used for the in vitro fertilization, and offspring has a different phenotype between the space flight group and the ground control. So there is some sort of like a transmission of the space experience stress. And now the question is if this is the case for the human, in human, there are many different factors, gravity, radiation, and other factors, which might be important for these causing these changes, and then mechanisms and biological implications. So to answer to these questions, we do lots of genomics analysis. And one prominent example is RNA sequencing, measuring the gene expression. By doing the sequencing analysis of RNA and complementary DNA, we can measure which gene is expressed, how much copies, and then comparing the ground control, keeping in on the Earth and the space flight, we can see the difference of the gene expression using RNA sequencing. So that's a major activity of the lab these days. And then, of course, the bioinformatics analysis to identify which genes, which category of the genes are affected, and then biological process. So our main goal subject is how about it in human. And then, as you may know, you can imagine the mouse experiment, we can dissect many tissue out, but astronauts is a healthy human, and then it's very difficult to get the biological materials from human. And then only thing we can potentially get is maybe blood sample, plasma, and also some urine or sweat or the other type of the natural material expelled from human. And then our target of the analysis is the liquid biopsy, the plasma cell-free RNA and DNA analysis. And again, this is also quite common in the cancer field these days. We already know that dying cells or produce a lot of the cellular debris which go into the bloodstream. And there are also many extracellular vesicles containing RNA released into the bloodstream. So our idea, which is not very special, but what we are trying to do is to analyze this extracellular vesicle in plasma samples to identify the molecular change in the internal tissue in astronauts. So doing experiment, clinical research, particularly in space, is a very long process. We need to go to the ethics committee, planning, and also approval, and also technically we need to validate lots of things. But these recent 1-2 years, we finally obtain the data and then do the analysis result. And then today I'm going to introduce some of these analysis results in this talk. This is a kind of typical scheme of the space flight experiment, but in our cell-free epigenome study, we recruited the 6 astronauts as a participant, and we collected blood samples at 11 time points, like pre-flight, before lunch, and then in-flight, staying in the International Space Station, and then coming back, and then readaptation to the ground situation. And then we collected blood samples and analyzed plasma RNA and DNA. And this is an example of we performed different type of the RNA sequencing and DNA sequencing, but this is one example of the RNA sequencing, cell-free RNA. As you can see here, this is a PCA plot, kind of like a dimension reduction to show the relationship among the samples, the before flight and then in flight and then post flight samples. So as you can see here, so the red dot is quite scattered around. So that means on the ground, different astronauts are very different from each other. But interestingly, once they go into the space flight, packed into ISS and then experience kind of like similar environment, although they should be coming from the different flight, they all behave in the same way. And then after coming back from space, again, like a majority of the samples coming to this area, that means they respond in a very consistent manner towards the space environment and also during the readaptation to the Earth condition. So this helped us to identify the differentially regulated genes by RNA sequencing. This is a scatter plot showing, so as I just mentioned, the pre-flight, when astronauts are all living in the ground, they behave very differently. So the analysis of the pre-flight to flight was not very successful. You cannot see very clear-cut differentially regulated genes, but once we compare the flight and post-flight, there is a very clear distinction between the the situation in the space and also after coming back to the Earth. So one thing we can see prominently is the mitochondrial genes, which is kind of like clustered here in like a straight line. And as you can see here in this plot, the pre-flight and then during the flight and post-flight, mitochondrial RNA is actually increased in a plasma sample during the flight and then come back to the opposite side and then come to the kind of baseline after the flight. So this means during the space flight, the human body reacts to expel the mitochondria into the bloodstream. And this observation is supported by mitochondrial DNA analysis. This is by real-time PCR. Compared to this ground control mean as a 1, there is a significant increase of the mitochondrial DNA in plasma sample. So by looking at RNA and DNA, this result supports that there is an increased release of the extracellular mitochondria into the bloodstream. So we don't know the reason for this yet, but this is clear-cut kind of biomarker seen in the space. And other than mitochondria genes, there are many differentially regulated genes in space. And this is another analysis called a kind of pathway analysis, looking at the enriched term of the altered cell-free RNA. So the gene ontology term enriched in this differentially regulated RNA is maybe first thing is the oxidative phosphorylation. This represents lots of mitochondria genes. and also some vitamin D receptor signaling, synapse, nervous system development, and also some other neuronal, and also some ionic the mineral metabolism functions seen in this gene enrichment analysis, and indicating the non-blood cell type RNA. This indicates that successful detection of the internal tissue change by just taking the blood samples. And this is tissue specificity analysis. Cerebellum, the part of the brain, is enriched, and the skeletal muscle, stomach, retinoblastoma. So this non-blood type tissue type enrichment confirms that the internal tissue RNA coming into the bloodstream, and we can detect in astronaut. And these RNAs are changing during the space flight. So just some of the example, for example, like cerebellum related genes. These genes are typically in a pre-flight as one, and then during the flight, day 3 to 120 tend to go down. And then once astronaut came back to the space, the Earth, these RNA going up. So it's an opposite reaction. And then seemingly coming back to the baseline after about 3 months or 4 months of the stay on the Earth. And interestingly, some of the genes really is tissue specific. This is an example of this CACN1A gene. This is GTEx database, this human gene expression database. As you can see here, this is a close-up of this area. So the brain, cerebellum tissue has a high expression of this gene, and essentially no other tissue type expressing this gene. So this is a very strong indication that we are detecting the molecular changes or RNA derived from this brain part. Just one more example of the skeletal muscle. This differentially regulated genes, sometimes genes increase the expression and going down after the flight, or going down during the flight and going up after the flight, and then again coming back to the kind of baseline level after 4 months of the flight, post-flight phase. And again, this RYR1 gene, for example, is highly specific to skeletal muscle. This also indicates this liquid biopsy, like taking blood samples and analyzing cell-free RNA, can be useful to detect the molecular changes coming out from the skeletal muscle tissue. So based on this analysis, we repeated this kind of GTEx database analysis in a bulk. And this heat map shows many tissue types and also the differentially regulated genes and tissue enrichment. So as I mentioned, cerebellum specific gene is clustered here. So the red color means across this same gene, different tissue row, this is the highest genes expression. So this indicates specific to cerebellum. And also this is a spleen. This is like artery, the heart, and this area is indicate adrenal gland and so on. So we can see many mixed profile of cell type tissue, cell type specific RNAs. That means by taking blood samples, we are detecting the mixture of many molecular signals from each individual gene, tissue types. So coming back to this, the kind of like a mouse observation confirmed in human, it's a general response, space response in many different tissues. So the next question we asked is, this is a gravity effect or not? So to do this, to investigate this point, we have very useful equipment in the International Space Station in a Japanese experiment module Kibo. This is a scheme of how this mouse experiment was performed. So in this International Space Station, we have this kind of mouse cage chamber, and then one part keeps this mouse cage stationary. So in the space, it's microgravity. And then another part, like a bottom shelf, is a rotating platform. So this is like a circular shape, so rotating platform. Because it's rotation, we can create the artificial gravity based on the centrifuge force. So by using this equipment, we can compare in the same space station environment, atmosphere and air pressure and noise and so on. We can compare this artificial 1G gravity against microgravity in the space.
Ini bagian farmasi yang terlalu teknologi.
So this is a movie of the mouse kept for 35 days in the space. So this artificial 1G control, this is actually rotating platform. So mouse appear to be standing up on the floor plate because of the gravity. And then here, the microgravity, just like astronaut flying in the space, this mouse sometimes floating around like this, and they still keep eating and drinking the water and food, and then kept in a healthy condition. So we, when mice came back to the Earth, we dissected and then analyzed many tissues, mainly by RNA sequencing. This is a CT image of the bone. You can see here, it's almost empty inside of the bone, which is a significant bone density loss in space. And again, we prepared we collected blood samples from these mice, came back from 35 days flight. And then this time we have ground control and also artificial gravity control. So hopefully we can detect the pure gravity effect by comparing this 1G to micro G conditions. So this is the heat map of the differentially regulated genes. And then we confirmed this mitochondria RNA in plasma, which is extracellular mitochondrial release, is affected by the gravity. So by comparing this artificial gravity to microgravity, we could detect the significant change and actually increase during the space flight group. So this is consistent with the human data. And then we further confirmed, we compared the mouse differentially regulated genes in a microgravity to the ground control and artificial gravity to microgravity. So this is indicates a pure gravity impact. And then we performed the human disease pathway enrichment analysis. And then importantly, this category contained many human disease category, but about half of this category was also common in human. If we convert the mouse gene into human gene homologue, and then do the same analysis. So this strongly indicates that many, at least this part of the human space flight impact change is related to the gravity component. And this gravity, this common part is has many human disease pathway, but to simplify it, we made a word cloud for each term, and then we see this visual optic, including this mitochondria and muscle as expected. So this broad type of tissue type are affected by the gravity change. So this indicates many observation in the mouse are also the case common in the human, and also this gravity impacted part is particularly conserved between human and mouse. So then what is, this kind of different events, what is molecular drivers? And then we identified potential kind of like a systemic regulators of these many events. And vitamin D is one of these important molecule, also changed in the human mouse. Some of the transcriptional co-regulators expressions are going up or down, altered in the space during the space travel. And then because of this vitamin D receptor signaling or thyroid and parathyroid hormone signaling is very important when during the evolution from the ancestors of vertebrates to the amphibians, like strong bone and muscle. And this actually, the evolution from the ancestor to the amphibian is also related to the other type of stress response that are commonly seen in space too, like high oxygen, metabolic change, and circadian rhythm, and also UV protection above water. And also in space, the skin, lung, and stomach, intestines are also responding. So this type of extra skin, epidermal skin tissue are also changed during the space. So to explain this comprehensive change, we came up with maybe this space-related change is related to the functions gained during the migration from the sea to the land. So in essence, we for like by taking long time, the vertebrates evolved from the sea to land, and then finally we reached the space to ISS and then achieved the long-term stay. But what we found in space was actually human body and mice just going back to the sea, losing the a systemic failure of this evolutionary gained function during the sea to land transition. So this is kind of like an interesting idea. And then this also noted us that maybe evolutionary gained function may not be so stable. And then currently we are working on this kind of like working model. We divided this kind of adaptation of the evolutionary traits, which is the fitness to the ground, could include the evolution part and also adaptation part. And then evolution is, of course, genetically established traits. And then adaptation is continuous response to the environmental signal. In this case, this is gravity. And then once we go to the space, this gravity is lost. And then suddenly this adaptation, which is maintained by the gravity signal, might be lost to this evolutionary kind of old phenotype, which is a throwback phenotype, or like representing the the ancestor of the vertebrate in the sea, like less bone, less muscle, and also weak defense in the skin, and also not coping very well against the high oxygen concentration and air pressure. And this idea is actually potentially related to the many situations in which the ancient phenotype appearing, like Haeckel's recapitulation theory. During the early development, there is early evolutionary old traits comes out, but this could be explained by unmasking of the adaptation during the early embryogenesis, and then essentially representing the kind of like a naive form of the ancestors. And then based on this idea, we are currently working on this quantification and modeling of the adaptation. So by vectorizing the different phase of the evolutional environmental pressure and also adaptation response in biological regulation. And then using this idea, if we can model the environmental situation to the adaptation of the vertebrate or like many creatures, organisms, we could potentially model the future prediction and perform the future prediction, like what if the environment changed, how this life can behave, and then continue through the adaptation. So this idea is very important when we think about what's going to happen if we live on Mars, which is partial gravity, or stay in a space station in a microgravity for a very long time or multiple generations. How human and ecosystem on the Earth could imported, exported, and evolved on the different planet or in space stations. So that's our current research topic, and then we are studying this mechanism to expose ancient phenotypes. And then we are working on the epigenetic side, but some of the other people working on this are using AIs or different model system from wet and dry site. And the Japanese team forms different anatomy and developmental researcher, and also some researcher taking care of the stress response. And I participate as the providing systems biology side for this team. And then this space adaptation and evolution research theme is ongoing as an open science theme throughout the international collaboration. Okay, so that's was the research topic, and then this is our lab member, and then the mouse researchers in Tsukuba, and this is funding and acknowledgement. Thank you very much for the attention, and we still have about 10 minutes, so that's about Q&A.
Okay, thank you very much, Professor, for a very interesting, inspiring, very intriguing. It's like another world research. Okay, we're going to open the Q&A sessions, the second sessions. There's one question in the chat box. So in your opinion, what is the biggest bottleneck or challenge in genome sequencing and bioinformatics field these days? For example, Human Genome Project, even if it was deemed completely, but actually only 92% of the human genome is mapped. What is the biggest challenge in this project in your opinion?
Okay, that's very good question. So that's kind of related to the previous question, like how to come up with some meaning of this like a metagenomic analysis. So this question also mentioning about this rest of 8%. It can be important, but we don't know. So and also because this 8% is left after all the effort of this research. So I think a very important thing is to find the meaning of this, if this 8% is very important or not. So it's very likely that many different unexplained traits might be linked to this 8%, or it's also possible that this is totally junk DNA, and then 8% clarified or not may not impact so much. So that's like the finding the value of this 8% is, I think, the biggest challenge. If people believe this is very important, many researchers will come and then develop the technology, and then I think it will be clarified. Like a space research too, right? Going to space and then people can stay for 1 year, but why it's important? Then suddenly, if we think about going to the Mars, this is the biggest bottleneck in the space development. Human cannot survive too long in a space environment. So now we found like a sudden awareness of significance of this space life science, and then we are doing lots of research these days. So finding motivation is very important, and if it's particularly if this is something like a previously neglected field, you can be a pioneer of that research field, I think.
Okay, thank you for Professor. Is there any other questions from the participants? Okay, so while waiting for the questions, it's really interesting that the space research, it's like it's unimaginable in Indonesia. It's like but Japan, of course, is a very developed countries, and maybe you have the facility and all, but I'm wondering what was the ultimate goal. I mean, like, for example, because maybe in the future, you want to search for another world for human to live in, you know, like in the other planets or in the space, or just like to help the astronauts to understand the clinical changes, you know, what can we do to avoid those clinical feature, that unwanted clinical feature in the astronauts.
So that's a very good point. So I think even in Japan or like in the US, why we need to sustain this space research is a big argument. It costs a lot of money. So of course, one motivation is to send people to the Mars, Moon and Mars, but earliest possible schedule of the Mars, human exploration is probably within 10 to 20 years. So we are working on like a very long-term goal, but to sustain, practically to sustain our research activity, we need lots of like come up with different type of like, the explanation. One thing is like a bone and muscle loss is a very prominent problem for the aging population. That's one explanation. Okay. And we recently are thinking about why it's, this has an evolutionary perspective, which is a very, as a mankind, it's very important question, where we are coming from and where we are going. And then that sort of extra problem questions will enhance the research activity and also allows us to continue this kind of research activity. And also for the genomics facility, we take care of the space research, but as you can imagine, like a space sample comes down only like once every few years or several years potentially. So it takes long time. So maintaining like a consistent data quality and platform is maybe one of my duty to take care of the, often like take care of the clinical sample analysis, corona samples, research samples, all these resources, gathering the analysis, like the order users to maintain like a facility keep running is also important effort. And one day, like before corona, it was purely the research purpose, but suddenly like all the in the same set of the equipment and automation system suddenly become useful for the corona pandemic response. So sometimes maintaining the research activities has a kind of secondary use, and that's something we learned for this during this like past few years.
Okay, thank you for the explanation, Professor. We have another questions. Is it okay, Professor? It's 4 o'clock already.
Yes,
so
yes.
Okay. I think it's related to the previous talk. So thank you from Ronald Harrison. Thank you for the presentation about the concept and process behind the genomic analysis. Is it possible to use the same concept for genomic analysis to detect and understand the genomic informations of foodborne pathogens contained in food matrices?
So food matrices probably means like materials, I think. So as I just mentioned at the beginning, like a nice thing about the genomics analysis is it's all common, whatever the creature using the RNA and DNA, we can analyze in a very similar way using the same concept. So it's certainly possible. And then once you learn, like maybe the initial intention is for the food type analysis, but that same concept and technology is used for clinical research or clinical practice too. So it's like as a kind of like a general platform or basis of the medical technology or medical or like an industrial platform. Genomics is, I think, very important. And also that's why like I'm also working for the lab testing companies, supported by the company, and also robotic company. So that sort of like a social demand is also important.
Okay, yes, thank you, Professor. Is there any questions? Other questions? Okay. Yes, Mr. Ling Nguyen Long.
Yes, you can call me Ling.
Okay.
So there's a natural occurrence that island species would evolve completely different comparing to their mainland counterpart in terms of size or bone structures. So would the evolution changes that be that drastically if humans have a Mars colony and procreate through multiple generations, and can those that live in Mars live in the same conditions on Earth?
So that's a very important and interesting question. We also like to answer. So based on, to our surprise, this, strong bone and strong muscle and also, intact like a body function, homeostasis, let's say, is, we thought this is very established evolutionary trait. And then if you see, if you think about, living on the Mars, maybe we are only thinking about maybe like several hundred years or tens or 20 generations only, but classical like Darwinism, let's say, the genetic mutation and selection does not fit to this kind of very short time range. So based on that, like a thinking or like a note, sudden change of the very important function in space was very surprising. That's why we came up with the maybe like a evolutionary acquired function, maybe like a big portion than we thought was depending on the adaptation and also intergenerational impact. So it's possible that relatively in the short term, people living on Mars for like multiple generations could be completely different appearance, like alien, big eye and green skin or something like that. So that's something we are proposing, future prediction.
Okay. Thank you, Professor. So I think I personally, I still have a lot of questions and also maybe the participants, but unfortunately we are running out of time. Once again, thank you for Professor Muratani for the really intriguing, like a new research, like new world for me, honestly. There's many new knowledge that I got from your talk today. So once again, thank you for the delightful presentations and also discussion. And now I will return this to the MC.
Thank you, Dr. Donna. And once again, thank you, Professor Muratani, for the insightful presentation. For your information, we have sent the attendance list as well as the evaluation questionnaire in the chat box. So please, for all of the participants to fill them out. Next, for a token of appreciation, the committee would like to give Professor Muratani and Dr. Donna an e-certificate. The certificate will be given virtually by Dr. Apotheker Sutrio, Magister of Science, as the Vice Dean for Resources, Venture, and General Administration, Faculty of Pharmacy. Please, Pak Sutrio, to give a token of appreciation for Professor Muratani. Okay, please give a virtual applause for Professor Muratani. Thank you.
Thank you.
And Pak Sutrio?
Yeah.
Oh yeah, okay.
Okay. Sorry. Thank you very much, Mas Afumi, for your lecture in Faculty of Pharmacy. We have certificate for Mr. Mas Afumi for lecture in Faculty of Pharmacy. Thank you. Sorry.
Okay.
Thank you very much.
Thank you very much, Mr. Mas Afumi.
Thank you, Pak
Sorry.
Sutrio. And also for the moderator, we have a token of appreciation, a certificate for Dr. Donna. Please give a virtual applause.
Thank you.
Okay, thank you very much. Well, ladies and gentlemen, now we are going to take pictures for documentation. So we encourage you all to please turn on the video and please one of the committee to guide this photo session.
Okay, I'll guide this photo session. So please open the camera and pose. Okay, 1, 2, 3. Okay, next slide. Okay, 1, 2, 3. Okay. Once more. 1, 2, 3. Okay, thank you very much.
Thank you.
Thank you.
All right, ladies and gentlemen, again, we would like to remind you all to fill the attendance list that we sent in the chat box. And for your reminder, the next lecture will be held next week, Tuesday, 23rd of August, 2022, with 2 lectures, consisting of lecture number 3 from Professor Dr. Tranti Tui, School of Chemical Engineering, Hanoi University of Science and Technology, that will start at 10 a.m. Jakarta time, and lecture number 4 from Professor Dr. Nguyen Tuha from School of Chemical Engineering, Hanoi University of Science and Technology, that will start at 1 p.m. Jakarta time. And the Zoom will be open 30 minutes before the lecture starts. All right, finally, we come to the end of today's agenda. Hopefully all of us get the value and benefits from the lecture. Thank you very much for your kind attention and participation in this summer course. I'm Hindun Rizni, signing off, and see you next week. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you very much.
Thanks, Prof.
Thank you,
Muratani.
Muratani.
Thank you, Muratani Sensei.
Ya, Nuriza.
Iya, Bu.
Mana Nurisa sehat?
Alhamdulillah sehat, Pak. Farmasi gimana kabarnya?
Baik-baik saja.
Ditunggu di sini ya, Pak. Di Sukuba.
Mudah-mudahan bisa ke sana.
Iya.